This is part one of the recorded lectures on cell division. This chapter is going to include the processes of mitosis and meiosis. So kind of like the chapter on metabolic pathways, it's really one unit which contains really the material for two chapters. So there's a lot of information in this unit on cell division. So why do cells need to divide? Cell division is necessary for many reasons. It's necessary in unicellular organisms. It's necessary for reproduction because unicellular organisms, which are composed of only one cell, have to divide in order to reproduce. In multicellular organisms, it is necessary for growth. To grow, your cells have to divide and you have to gain more cells, a greater number of cells. And for injury repair, your body has to produce new cells to replace the ones that were lost or damaged. When cell division goes awry, meaning when the cell division process doesn't work, dramatic results may occur. Without sufficient cellular oversight, repeated rounds of unregulated cell division can lead to minor conditions like psoriasis, which is, a skin disorder, or a life-threatening disease like cancer. So it's very important that cell division work properly. Cell division takes, that's weird, the way it's worded. Cell division occurs by a strict cycle with multiple stages and checkpoints to ensure that things don't go awry, that things don't get messed up with cell division. Okay. DNA, of course, deoxyribonucleic acid, is the genetic material of living organisms. So if a cell is going to divide, it has to first replicate all of its DNA so that the two new cells, which we call daughter cells, will get an exact copy of the DNA of the parent cell. So DNA provides the instructions or the genes for organisms to grow, to function, and to respond to their environment. When a cell divides, it will pass on a copy of its DNA to each of its daughter cells. So what that means is it must replicate its DNA. It must make a copy of its DNA in order to pass on a copy of it to each of its daughter cells. DNA is also passed on at the level of organisms, with the DNA in sperm and egg cells combining to form a new organism that has genetic material from both its parents. So basically, here's what we have. If let's say this is there we go. Let's say this is the parent cell. For example, we know our skin cells have to be replaced periodically because we lose our top layer of skin every so often, and those skin cells have to be replaced. So let's say that this cell that I just drew is a stem cell that produces new skin cells. And we know that we have a copy of 46 chromosomes, which contain our genes. Our DNA is in the form of 46 chromosomes in humans, and that contains our genetic information. So when this cell divides and makes two new cells, which we call daughter cells, they need to have 46 chromosomes also. And that can't happen unless this original 46 copies itself. Okay? Now, whenever you have a sperm, that sperm only has 23 chromosomes, half the DNA of the dad. And when you have an egg, that egg only has 23 chromosomes, which is half the DNA of the mom. So when they unite and form a zygote, then the zygote has 46. So a little bit more about DNA in eukaryotic cells, cells that have a nucleus. Most of the DNA is found in the nucleus, but we also know that chloroplast and mitochondria also have their own DNA. But primarily what we talk about when we discuss cell division, we're really just talking about the nuclear DNA, really. But everything inside the cell has to be copied so that the new cell can have a copy of everything that was in the parent cell. So that would include the organelles, it would include the mitochondria, the chloroplast, the Golgi body, the endoplasmic reticulum. Everything has to be copied so the two new cells can each have their own copy of everything that was in the parent cell. Okay, so in prokaryotes, the DNA is not housed in a membrane-bound nucleus. It is just found in a region of the cell, and that region is called the nucleoid. But there is still DNA in prokaryotes or bacteria and archaea. Now, the set of DNA in a cell is called its genome. So all the DNA in a cell is called its genome, because all the DNA means that that is all of the genes within that cell. And all the cells in an organism, with a few exceptions like egg and sperm, contain the same DNA. You can also say that an organism has its own genome, and since the members of a species typically have similar genomes, you can also describe the genome of a species. In other words, I'm an organism, and all of my cells except for my eggs have 46 chromosomes. I don't know. I'm trying to think of a person. Oprah Winfrey. Oprah Winfrey is another human from the human species, from the Homo sapiens species, and all of her cells, besides her eggs, have 46 chromosomes. That's the number of chromosomes in each of our cells. So our genome is composed of 46 chromosomes, half which came from our mother and half came from our father. Now, chromosomes are rod-shaped structures that look like these structures that you see here. This is called a karyotype. This is a human karyotype showing the collection of chromosomes in a cell of a human, and the green chromosomes are from the father, the purple chromosomes are from the mother. And so you can see that they come in pairs. They come in homologous pairs. So there are 23 total pairs of chromosomes. Pair number 1, pair number 2, pair number 3, all the way down here to pair number 23, which in this case is an X and a Y. A Y is like half of an X, really. It's just called a Y chromosome. But an X and a Y means that this person, this individual, is biologically male. So this also shows you that the cells of most human cells and most of the cells of multicellular organisms are diploid. That means they contain 2 copies of each set of chromosomes, and we call them homologous pairs. So in humans, we have 23 homologous pairs. The 2 homologs have the same type of gene in the same place, but they can have different versions of genes. Versions are alleles. So let's say, for example, let's just say for pair number 15, okay? Let's say right here and right here on each chromosome is a gene for whether or not one human trait is whether or not you can roll your tongue. We'll get to that later in genetics, but it's the first one I thought of. So let's say that it's the gene for tongue rolling. Well, there's two alleles for that gene. There's the big R allele, which means you can roll your tongue. You are a roller. And there's the little r allele, which means you are a non-roller. Now, the gene for tongue rolling is going to be located in the same place on each chromosome, but it doesn't have to be the same version of the gene. Both genes can be for big R's or for roller. Both can be little r's for non-roller, or you can have one of the genes that's a big R and one that's a little r. The point is, it's genes of the same type, but they do not have to be the same version. You know, lots of different human traits like hair color and eye color, are not the same in the mother and the father. So the genes for those traits are located in the same place on the chromosomes, but they just don't have to be the exact same type. So in humans, the X and Y chromosomes, like we said before, determine a person's biological sex. Two X's means the person is biologically female, and an X and a Y means the person is biologically male. While the two X chromosomes in a woman's cell are genuinely homologous, the X and Y chromosomes of a man's cells are not. So all these chromosome pairs, all the way up to from pair 1 to pair 22 in a man, are called homologous chromosomes. Homologous chromosomes. But you cannot call the 23rd pair a homologous pair of chromosomes because one is much smaller than the other and doesn't contain the same set of genes as the other one. So the X and the Y chromosomes are known as sex chromosomes, and then the other 22 pairs, or the other 44 total chromosomes, are called autosomes. So there's 2 sex chromosomes and there are 44 autosomes, which makes 46 total chromosomes in a human cell. All right, so how DNA is organized. This is just the DNA double helix, which is just naked DNA. What happens is inside the nucleus is DNA wraps around a histone protein. The histone protein is all this inside here. It's a large histone protein. So DNA wraps around a histone protein, and then a lot of DNA wrapped around a lot of different histone proteins coil up into a structure called nucleosomes, and then those nucleosomes coil into chromatin. And chromatin is very, very spread out. It's not condensed into these shapes that we can see, like in this karyotype. These chromosomes are rod-shaped. If it was chromatin, it would be very, very spread out chromosomes. Just to give you an example, in the nucleus of a cell, this is the nucleus, not the entire cell. So in the nucleus of a cell that is not dividing, its DNA is going to be all spread out, so much so that the nucleus under the microscope will appear a solid color. Okay, and then in the nucleus of a cell that is about to divide, the chromosomes will appear rod-shaped, or really X-shaped, because the two rods will be divided in the middle, and you will see them, they will have condensed into chromosomes like this right here. Okay, and then this is just the intermediate stage between chromatin and a duplicated chromosome, or a condensed chromosome. Okay, so now we'll talk about phases of the cell cycle. The cell cycle is divided into stages of growth, DNA replication, and cell division, and it's going to produce 2 identical cells or clones of the parent cell. And we're going to discuss interphase and the mitotic phase in the next lecture.